Okay. Am I audible?
Yes, sir.
Yes, sir.
Okay. Thank you. So we can start, right? It's 12:12. Okay. Can you see my screen?
Yes, sir.
Yes, sir.
Okay. So you say that we have discussed pairwise alignment. So we have discussed that how alignment can present the similarities and dissimilarities in between two sequences. Just a minute. Can you allow who are actually joining? Is it possible for you? Are you getting the function?
I allowed the.
Okay, okay, please allow, okay, on behalf of me.
Okay, sir.
Okay, sorry for the inconvenience. So we are discussing about alignments. Alignments is an efficient tool that can mark the similarities and dissimilarities in between two sequences. Here we are talking about only pairwise alignment, not multiple alignment. We will discuss it later on. So the similarities and dissimilarities are actually associated with the functional similarities in between two genes or proteins. So in that context, we have seen that Needleman and Woons, they took the effort to design an algorithm that is fully mathematical and statistical algorithm, and which can successfully find out the similarities in between two closely related sequences. Sorry, why we are talking about two closely related sequences? Because we have seen that as the sequences diverge, the dissimilarity increases. Now, if the dissimilarity increases, then there is a chance to miss the biologically functionally similar sequence, which are actually mathematically becomes dissimilar. That is the problem. I repeat the point again. This is the beginning point of our discussion today. What happening if sequences are diverging? That means two functionally similar protein, because of species diversification, they are becoming a distantly related organism source. So source organisms becomes distantly related with respect to the taxonomical position. Why? Because of species diversification. Earlier, they were very close, maybe they were present in the same organism, but after thousands of years, okay, the organism has diverged, and now the same protein, they belong to two different organisms which are taxonomically at a distant position. Are you getting my point? Yes or no?
Yes.
Okay. Now these two protein, they will be functionally similar, but they will accumulate number of mismatches. That means the dissimilarities will increase in between these two sequences. Now, if we are following Needleman and Woon's algorithm, the global alignment method, mathematically, they may consider these two sequences dissimilar because they follow a cutoff value of their score. The score you have observed in the last practical session. So if there is a cutoff score, they will consider that these sequences are dissimilar mathematically, but biologically we have seen that they are functionally similar. So then Smith and Waterman, they thought that how? we can actually retrieve these sequences which are becoming mathematically dissimilar in Needleman's algorithm, but biologically they are similar. So he started thinking about the patterns in between functionally similar proteins, but they are from diverse organisms. And what he found was that the similarities among the functionally similar proteins, they are localized in a particular position. That means at some particular position, say there is a protein of length 1 to 500 amino acids. Now in 500 amino acids, you may observe that say from 10 to 50 there is highly similar region, then 51 to say 100 that is dissimilar, then 100 to 250 regions that is highly conserved, again from 250 to 300 there is dissimilarity, the rest of the regions are highly similar. That means you have three highly similar regions, in between there are some dissimilarities. Now if you align these sequences by using Needleman wounds, their score will be down, and the problem is that they are biologically similar. That means Needleman wounds algorithm is having the disadvantage that it is not getting the functionally similar proteins from diverse organisms. So in that case, to focus on the local regions only, Smith and Waterman proposed local alignments. Why local alignments? Because it was observed that these local regions are responsible for function. How? If you mutate these organisms at these positions which are conserved, you will observe that the proteins, they lose their function. Whereas if you make mutation in the non-conserved regions in this protein, you will see that their function remains intact. So that thus it was proved that the conserved regions were responsible for getting functional similarity. Now Smith and Waterman actually they did not design a new algorithm. What they did, they just modified the NW algorithm. How? You can see here there are two differences. So if we go through the difference, it will be easier for us to understand and remember NW and SW algorithm both. So now see that what are the two differences in scoring methods. If you remember the third, the three ways we considered for Needleman-Wundt scoring, either diagonal path or horizontal path or vertical path. Out of these three paths, whatever value you are getting, the maximum value was considered for a particular cell. But here, Smith Waterman, they just added a 0. That means now there are 4 values from this 3 path and 0. Which one is maximum? That will be the value for that particular cell. Okay, so there is one difference. The other difference is that the alignment can now end anywhere in the matrix. We will see while we are discussing the algorithm. That means earlier in Needleman-Wundt, the alignment was starting from the last cell and the alignment was continued till the first cell. So that's why it was a global alignment. So now because of 0, introducing 0, now the alignment can end anywhere in between the path. How? We are going to see that shortly. So far so good? Yes?
Yes, sir.
Okay. Others, are you getting?
Yes, sir.
Okay. So now let's take consider the same example we discussed for Needleman wounds. Take the same sequence, see the sequence 1 and sequence 2, same scoring parameters and same gap penalty. Why we have kept it same? We want to see the difference in Needleman wounds and Smith Waterman so that we can understand it very clearly. Fine. So let's go ahead. See, if you follow the score calculation by Needleman wounds, you got various positions minus marks, minus score, isn't it? But you know that in Smith Waterman, there will be no minus value. Why? Because in the principle, you have introduced a change. What is the change? There will be a 0 along with the 3 path. So out of these 4 values, which will be maximum, that will be considered for that cell, right? So that means in Smith Waterman score calculation chart, there will be no negative values. Okay? Because always if there is negative values from these 3 path, then 0 will be maximum. So that's why there will be no negative values. Now, like the Middleman rules, all the other rules and regulations are exactly same in case of Smith-Waterman algorithm. So now this scoring matrix, after the calculation of cell wise score calculation, it looks like this. The matrix look like this. Okay, that was quite different in case of Needleman rules, right? Are you getting?
Yes, sir.
Yes.
Okay, so I will ask you to actually calculate the scoreboard of Smith Waterman also, and please uh, show me it in the next session like NW. Okay, so this two scoreboard, please get it signed by me. Okay, so now in Smith Waterman there is another change. What is the change? The change is that we will start the alignment from the highest cell value. In Needleman rules, what we did? We considered the last cell, whether the value is maximum or not. We are not bothered about that. We had to start the backtracking from the last cell. But here, the Smith-Waterman algorithm, they said that no, 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 no. You are not confined to the last cell only. You have the freedom to consider the cell which is having the highest value. So please look into the cells where we have the highest value. You see that this is the highest value, right? The 28 is the highest value in this scoring matrix. Can you see that? Is there higher
Sir.
value other than 28?
No, sir.
No. So 28 is the highest value. So we will start the backtracking from this cell. And the rules are same for backtracking as it was in case of Needleman rules. So as per the backtracking rules, now 28 encompasses the cell E by E, right? So let's align E by E because we are from the diagonal path. We are from the diagonal path. So we had a rule that if we are coming from the diagonal route, diagonal cell, then we will align the respective cell. If we are coming to that particular cell other than diagonal path, we will put a gap. Where to put a gap? Where the arrow is heading, in that sequence you will put a gap. So as per that rule, here we can align the cell E by E. Now let's move on. So now the next cell is 22, value 22, right? So here also we have come from the diagonal path, right? And now here we are going to align H by H. This is the cell, right? H by
Yes.
H. Now we are in this path 12. Okay, we have come from the last horizontal cell. So as per our rule, we are not going to align these two sequences. Rather, we are going to put a gap where in sequence 2. So let's align G by gap. Correct? Am I correct?
Yes, sir.
Okay.
Yes, sir.
Okay. Now, say you are in the cell value that is 20, right? So here, see, we have come from a diagonal path, right? So now we can align W by W. Okay, we can align W by W. Now say we are in this cell having value 5, and again we have come from a diagonal path. So here we can align A by A, right? And then finally you see that we have encountered 0. So now you have seen that there are two differences. I said in the earlier that Smith and Waterman alignment can end anywhere. So whenever you will encounter 0 from a particular path, you will end that piece of alignment at that position. That means this is our optimum local alignment. Now you can see that just putting a gap only in between these two sequences, we have retrieved a similar region. Can you see that? Okay. So now, as per the rule, we can start multiple local alignments. We can get multiple local alignments. To do so, we have to delete the regions around the best path. The best path we have just considered, right? We considered the highest value and we followed from highest value to 0. That was our best path. Now we have already created the alignment for that path. So we will delete that path and we will repeat the backtracking process that we have considered so far. Okay. So now see that we will delete this path, right? So now what is the next highest value? The highest value is say 21. Clear? So now we can start our next optimum alignment from the highest value after deleting the best track, best alignment track that we have deleted. So now it is a different track that is not at all linked with the previous track. That's why it is a two different local alignment within the same sequence. Within the same sequence, we have two conserved regions. They are not linked with each other. So that's why we are trying in that pathway. So now the highest value is 21. So as per the rule, we have come from the last diagonal path. So we will align A by A, right? We will align A by A. Then next we are in the cell which is having the value 16. So again we have come from another diagonal path. So we will align E by E. Clear? Then you see we are in the same cell having value 10. Again we are coming in the diagonal path, right? Then you will align this cell H by H. Now you are encountering the 0 and you can end the alignment. This is the second best local alignment. Now you can see the two best local alignment. The first, you have seen that the length of that local alignment was larger as compared to the second best alignment. Now you see, you consider the two sequences. Now you are in a position to compare when to apply NW and when to apply SW. How? you see that looking at the sequence 1 and 2, it seems the sequences are dissimilar. Okay, because it, if you analyze the sequence without any analysis calculation, it seems that it is dissimilar. Now if you apply NW, you have seen that you can get a similar region with many gaps. But if you apply SW, you see that just putting one position gap, you can find out two conserved regions. Now, if these two sequence, if you can find that they have similarity with respect to their function, you can analyze or you can conclude that these two regions might be the key region for functional similarity, and the wet lab option is open for you to prove that. Okay, so in this way, if you have the unknown sequences for which you don't know function, you can match or you can compare or you can align with the known, functionally known sequences, and you can determine its function by applying either NW or HW as per the taxonomical distance of the sequences. Any questions?
No, sir.
ওকে, তোমরা বুঝতে পেরেছো তো?
Yes, sir.
ওকে, তাহলে